Okay, next question. So I'm Dr. Ben Newman, and I study coronaviruses. And looks like this question doesn't even have a coronavirus in it, so I'm going to do my best. This is from Lisa. Hello again, Lisa. With the vaccine tech, the new vaccine tech against viruses seeming to work, and work really well despite a number of different approaches. That's really cool. Will this lead to vaccines being developed against other viruses such as HIV, Ebola, etc.? Yeah, okay. So, for Ebola, we have a couple of pretty good vaccines. Now, you probably haven't taken one because they are used, they're not vaccines that are given to everybody. They are vaccines that are used when there's an outbreak, and they're used in the area where there's the outbreak. And so Ebola tends to flare up in this band right across the middle of Africa, like where all the cool jungles and forests are and all the neat animals. I guess, no, there's some neat animals down on the plains, but you get what I'm saying. And so, it works pretty well. There are a couple different versions, and one of the versions looks a lot like the adenovirus vectored vaccine. So, like the Oxford adenovirus, there's actually a version of that that's using, instead of the spike, it's the Ebola virus equivalent to the spike. That's doable, and I have seen, I believe, experimental RNA vaccines against Ebola. I don't recall any of those being approved, like sort of widespread testing. It may be there in the literature, and I may be missing it. And if I am, I apologize to all the people that worked really hard on that. Sorry. I need to read more. But there's no reason why it couldn't work. And not just for those, for a lot of other viruses. If mRNA vaccination turns out to work as well as it has been working for coronavirus, this is potentially the way that you would want to make all vaccines going forward. It's just really easy to make a messenger RNA vaccine, because as soon as you have the sequence, and as soon as you know which protein sticks out of the virus, which we can tell based on what kind of virus it is, then you just put that thing in a messenger RNA, wrap it up in the same little lipid concoction, and boom, inject it, and it ought to work. There's not really a good reason why it wouldn't, except when we get to HIV. People have been trying a lot of stuff for HIV, and so here again, I don't remember seeing any straight messenger RNA. The glycoprotein is GP120 and GP41, or the envelope protein. I guess that's what regular people would call it. I don't recall seeing a large clinical trial of a messenger RNA vaccine there. The problem with HIV is that you have quite a lot of HIV-like virus already in your body. It's actually embedded in your DNA. The most conservative estimate is that you've got about 6 HIVs per gene that you have, like just in terms of total amount of coding capacity in your genome. So that's really a lot of HIV. There's something like 6% of your genome, and the actual genes that you express as a human to be human and build yourself, those are only, I think, about 1.5%. You can look at HIV related things, and if you look, a little bit wider search, that blossoms out to, in some cases you could say that roughly half our genome, or a little more or a little less, is basically HIV-like material. So there's a lot of that going around, and the problem is you have to train cells to attack just the HIV version and not any of these other fully fledged retroviruses that are embedded in your genome. Now, some of these retroviruses still have active proteins. Most of these are dead in some way. They're sort of genetically broken and you don't have to worry about them. But there are enough of them that are able to make some pieces. And one of the pieces is called syncytin. And it actually is really important. This is how babies are fed inside the womb, is with this ancient remnant of an HIV protein. It sort of fuses some cells together so we can transfer nutrients from mama to baby. So Yeah, the problem with HIV is really hard. And the other problem with HIV is that with a coronavirus, it goes in and it lives in your cell, but it doesn't become physically part of your cell. HIV is the opposite. It goes in, and not quite the first, but maybe the second thing it does is to turn itself into DNA, cut your DNA, open it up, glue itself in there, so it is now literally part of the genetic material of your cell. And from that point, it can just keep running off little copies of its HIV. You're going to smash most of these things, but it's in the genetic code. It has all the time in the world. It's just got to sit back and wait for that one day when, one white blood cell doesn't bother to do its job and patrol a particular area, and then boom, yeah. You get some virus coming out, you get escape, you get bad stuff happening. So HIV is a really tough out compared to these little RNA viruses like flus and most of your cold viruses, yeah, which doesn't mean anything scientifically, but I'm thinking of a great big basket of different viruses. And things like Ebola and, frankly, a lot of viruses out there. So I think the mRNA technology is really good. I have no idea how to solve the HIV sort of vaccination problem. We may just have to solve that one by reducing spread through sort of aggressive treatment of anybody who's got it to keep the virus load down and keep spread lower that way. And yeah, just sort of education, awareness, education. That's like, I have the knowledge and I will give you a small piece, yeah, because I'm so kind. No, it's just like awareness, yeah, knowing what this is, how it spreads, how it doesn't. Which, yeah. For people of a certain age, they probably heard it already, but there's these new people that keep coming into the world, and so you got to keep saying it because there's always somebody new that's just hearing it for the first time, I guess. So yeah, really good question. Yeah, sorry I don't have a better answer than that, but I think mRNA vaccines are here to stay and are the way forward very much, which is what the mRNA vaccine community has been saying for years. I don't know, 20 years, 30 years, so whatever. Yeah. All right, they were right. Yeah. That's all. Thanks very much. This has been Ask Dr. Ben.